Hi, I'm Zach Hall, co-founder and CEO of Prosel Biosciences. Prosel Biosciences is developing Seleno Express, a seleno protein engineering and expression platform technology in recombinant green algae. Selenocysteine is the 21st amino acid, and selenocysteine-containing enzymes and proteins have many desirable chemical properties, resulting in increased catalytic activity and stability under conditions of high pH or temperature. To date, selenocysteine engineered enzymes and proteins have not been economically feasible because only a few model organisms have the molecular machinery required to translate selenocysteine amino acid residues into proteins. One of those model organisms is green algae. Importantly, the ability to incorporate selenocysteine into proteins is a novel molecular tool capable of enabling significant functional improvements to molecules with existing multi-billion-dollar revenue streams. Two such multi-billion-dollar vertical markets include industrial enzymes for applications ranging from biomass conversion, plastic degradation, food processing, and biofilm surface cleaning, and for the pharmaceutical industry for pharmaceutical drug products and raw ingredients used in pharmaceutical manufacturing for biologics and small molecules. Procel's commercialization strategy is to file multiple patents on a per product basis, reduce those inventions to practice via pilot scale manufacturing, and seeking a suitable partner to outlicense or enter into joint development agreements with. Our products are proprietary strains of recombinant algae that express selenocysteine containing enzymes and proteins with high commercial value and applications at the commodity scale. Industrial enzymes and human therapeutic proteins alike evolved to surmount problems and challenges associated with a range of factors that limit efficacy, resulting in increased costs to manufactured goods. Prosel Biosciences will generate new intellectual property and licensed technology from Dr. Sayer's existing patent portfolio to metabolically engineer green algae to express molecular products with improved functional chemical properties for targeted commercial applications. Procel's Seleno Express platform will therefore solve inherent limitations with industrial enzyme catalytic activity and stability of pharmaceutical drug products circulating in the bloodstream. Overcoming problems and challenges of existing commercial products by designing a better molecule has been a proven formula for success and shown to translate sustainable growth and recurring revenue creation from commercial milestones, upfront licensing fees, and royalty payments from strategic partners, which are often large multinational corporations with global manufacturing capabilities. Today, Procel's initial product development candidates have a combined market cap valued over $30 billion. Procel's advantage is to pair subject matter expertise in computational bioinformatics from Los Alamos National Labs computational core with a broad mechanistic understanding of photosynthesis and decades of experience expressing recombinant proteins and purifying lipids produced from proprietary strains of green algae. Prosel has filed several patents to protect intellectual property involving protein expression, secretion, and purification of recombinant proteins. Our first human therapeutic protein candidate is our minimal viable product and was chemically synthesized to contain selenocysteine and shown to improve stability fourfold in vivo. The next two slides will focus on some of the unique attributes of selenoprotein engineering and green algae. As mentioned previously, green algae are one of a handful of organisms capable of incorporating the 21st amino acid, selenocysteine, into proteins using specialized ribosomal protein synthesis machinery. The advantages of selenocysteine include the pKa of selenocysteine being greater than the amino acid residue cysteine, which has a broader range of redox valence states, and a more negative redox potential than the sulfur atom contained within the cysteine. amino acid residue. You may be asking why this is important. Put simply, selenocysteine modified enzymes often have several thousand-fold higher catalytic activities than enzyme analogs with a cysteine as the catalytic site residue. This translates into improved functionality and applications for a range of commercial manufacturing processes. Engineering enzymes to have catalytic activities hundreds to 1,000-fold higher and pharmaceutical biologics to have increased in vivo stability in the bloodstream results from the selenocysteine amino acid residue forming diselenide bonds. This is in contrast to cysteine amino acids that form disulfide bonds, which can be easily reduced by a variety of endogenous reductants. Prosel has several product development candidates in discovery stage, and our commercialization pipeline includes proof of technology for selenoprotein engineered human therapeutic proteins and industrial enzymes. Prosel has an agreement with the New Mexico Consortium to occupy lab space and permit access to the Los Alamos National Labs supercomputing network. Access to the supercomputing network is an important piece of Prosel delivering on our value proposition to investors and strategic partners, and it enables targeted protein design to identify the right selenoprotein product candidates for vector development and commercialization. Seleno Express is economically competitive compared to existing recombinant protein expression systems and mammalian cell lines, and seen as a major benefit to express proteins from recombinant green algae. This is largely due to the cost of raw materials and the ease at which green algae proliferate under the right growth conditions, namely light, heat, and ionic nutrients. And purification can be readily performed using off-the-shelf ultrafiltration technology. Prosel's first human therapeutic product candidate, Pro756, has a market cap greater than $25 billion, and chemical synthesis of this seleno-modified peptide hormone has already been demonstrated to have a 4-fold increase in stability in vivo compared to the wild-type hormone. In contrast to mammalian-based recombinant expression systems for the same peptide hormone, green algae do not have any known human pathogens like viruses or mycoplasma contaminants. We anticipate the expense of protein expression from recombinant green algae to be very cost competitive with traditional mammalian-based recombinant protein expression systems. Through our relationship with the New Mexico Consortium, Prosel will have access to commercial scale photobioreactors and utilize advanced photobioreactor systems to demonstrate production, manufacturing, and purification of GLP and cGMP grade proteins from our recombinant green algal strains. Pro756 will serve as a proof of technology for the Seleno Express platform and is expected to be an early win in terms of identifying a pharmaceutical partner for out-licensing and commercialization. Furthermore, present estimates suggest that of the 359 million tons of plastics that are produced annually worldwide, 150 to 200 million tons accumulate in landfills or in the natural environment. An urgent need exists to develop a biological approach to addressing pollution from petrochemical-derived plastics. Analogs of Prosel's first industrial enzyme product candidate, Pro7384, has received a lot of attention lately for its inherent ability to depolymerize certain types of commonly used plastics. It has been shown that enzyme approaches to plastic pollution degradation exhibit the same properties as petrochemical-derived substrates and are readily recyclable. Reducing the degradation time to depolymerize plastics by improving the catalytic activity of selenoprotein engineered enzymes to depolymerize plastics in landfills addresses the plastic pollution crisis, and manufacturing of this commodity scale industrial enzyme to meet global demand would also benefit the environment via biological carbon capture and sequestration. With our commercial partners, we anticipate selling into the global plastics resins market, which has an estimated value over $680 billion in 2020. Products derived from Seleno Express platform include all manufacturing and purification methods, trade secrets, proprietary green algal strains, and novel seleno protein composition of matter claims, which represent the bedrock of Prosel's available licensable technologies to strategic partners across specialty chemicals, synthetic biology, and the pharmaceutical industry. Prosel has identified a number of potential strategic partners to outlicense our industrial enzyme and human therapeutic product development candidates, including Novo Holdings, DuPont, and BASF Chemical, to name a few. Prosel's discovery, development, and deployment timeline has begun with our first seed stage investment from Big Idea Ventures. We continue to raise seed stage funds from angel and early stage venture firms. We anticipate raising a Series A round within 12 to 18 months of launching lab operations and commercial product development. Prosel plans to generate revenue from private and public grant funding opportunities, sponsored research agreements, venture and angel capital investors, joint development agreements, and fees from out-licensed milestone and upfront licensing to strategic partners. Our founding team includes seasoned biotech entrepreneurs, Dr. Richard Sayer and Zach Hall, and computational bioinformatics expert, Dr. Wataru Nishima. We are building our scientific and corporate advisory boards, which include, to name a few, Dr. Shana Ivey from the New Mexico State University, and patent and corporate attorneys David Kerr and Jared Crane. If you'd like to learn more about what we are doing at Prosel Biosciences, please feel free to reach out to Dr. Sayer or myself. Thank you and bye for now.